A novel SARS-CoV-3 epidemic or pandemic genetic jump from SARS-CoV-2 and MERS-CoV has been forecast. Pause each illustration for a detailed study of the illustrations and data contained in this revised review of the literature. A. Animal, natural and intermediate hosts, origin of human coronaviruses. B. Clinical presentation of patients with SARS-CoV-2, including common, uncommon, and severe symptoms of SARS-CoV-2. C. Human coronavirus types, common human coronaviruses, alpha coronaviruses, beta coronaviruses, and other human coronaviruses. MERS-CoV, the beta coronavirus that causes Middle East Respiratory Syndrome or MERS. SARS-CoV, the beta coronavirus that causes the severe acute respiratory syndrome or SARS. And SARS-CoV-2, the novel coronavirus that causes coronavirus disease 2019 or COVID-19. D. Diagram of coronavirus virion structure showing genome and structural proteins, spike S, envelope E, membrane M, and nucleocapsid N. N protein forms a complex with RNA and aids in the viral assembly after its replication. SC and M proteins create the viral envelope. The S protein is a club-shaped surface projection, giving the virus its characteristic crown-like appearance on electron microscopy, which is responsible for viral entry into the human cell. Schematic diagram showing the replication cycle of SARS-CoV-2. The virus moves into the target cells via an endosomal path. Initially, S protein connects to the cellular receptor Angiotensin converting enzyme 2, ACE2. 1. The viral genome is set free. 2. And decoded into viral replicase polyproteins. 3. Then they are split into small products by viral proteinases. 4. Subgenomic negative strand templates are created from intermittent transcript on the plus strand genome and function as patterns for messenger RNA synthesis. 5. The full-length negative strand pattern is made as a pattern for genomic RNA. 6. Viral nucleocapsids are accumulated from genomic RNA and N protein in the cytoplasm. 7. Followed by a budding into the lumen. 8. Virions are then released from the cell through exocytosis. SARS-CoV-1 emerged in 2003 during a static period of exclusivity, yet undergoing genetic and structurally genomic-based evolution that lasted, culminating in the particular antigenic jump and emergence of the novel genetic variant SARS-CoV-2 in 2019. Sarbecoviruses, such as SARS-CoV-2, utilize angiotensin converting enzyme 2, ACE2, as a viral entry receptor. Merbecoviruses, such as MERS-CoV, similarly employ Dipeptidyl peptidase 4, DPP4, as its viral entry receptor. Certain human alveolar and small intestinal cells express ACE2 as well as DPP4, making possible co-infection by both viruses. SARS-CoV-2 is capable of utilizing certain of its ACE2 independent alternate receptors to enter host cells and co-infect with MERS-CoV in its cells expressing DPP4 and one of the alternative receptors. The result can be an emerging beta-CoV, SARS-CoV-3, or MERS-CoV-2 via the genetic recombination occurring between SARS-CoV-2 and MERS-CoV. Such beta-CoVs are capable of employing transcriptional, regulatory, and conserved sequences upstream of the genetic open reading frame mediating this continuous transcription of the viral genome, possibly participating in the recombination of SARS-CoV-2 and MERS-CoV into a novel pathogenic, highly communicable coronavirus in the form of SARS-CoV-3, causative of human morbidity and mortality of a severity and nature similarly related to that of its progenitors. Co-infection of SARS-CoV-2 and MERS-CoV may result in the emergence of recombined beta-CoV, SARS-CoV-3, or MERS-CoV-2, thus calling for the development of pan-CoV vaccine. A. Because of the identical infection cycle of SARS-CoV-2 and MERS-CoV, recombination may occur when ACE2 using MERS-CoV co-infect with SARS-CoV-2 in ACE2 expressing cells. 1. Or when SARS-CoV-2 and MERS-CoV co-infect a cell expressing both ACE2 and DPP. 2. Or when MERS-CoV co-infect with SARS-CoV-2 in the cells expressing DPP4 and one of the ACE2 independent alternative receptors. 3. Resulting in an emerging beta-CoV SARS-CoV-3 or MERS-CoV-2 via genetic recombination between SARS-CoV-2 and MERS-CoV. B. SARS-CoV-2 MERS-CoV recombination calls for the development of a pan-beta-CoV vaccine to prevent infection of the newly emerged beta-CoV SARS-CoV-3 or MERS-CoV-2. New presentations coming soon. Like, share, subscribe, and ring the bell for notifications. Thanks for watching. Thank you to current and new subscribers.